Hi, I'm Liz McDaniel. This presentation is for Chemistry 530, Concepts in Biochemistry with Dr. Kenneth Capps with Grand Canyon University, and this is all about G-protein coupled receptors. So just a brief overview to start off. Today we'll introduce you to G-protein coupled receptors. I'll talk about what they look like and how they work, and we will get into the locations and variations of GPCRs as well as their mechanism of action. And finally, I will touch on some of the mutations and their phenotypes, which result in GPCR-associated disease and also GPCR moderating drugs. Since there are so many variations of these receptors, I'm going to focus on some of the best understood and most common versions today. And like Maria von Trapp, I like to start at the very beginning. So I'm going to first talk about signal transduction. This is just a fancy term for how information is transferred from outside of a cell to inside a cell. Once a piece of information is transmitted into a cell, it can then respond appropriately, either by synthesizing a new protein, generating the release of small molecules, changing membrane potential, or continuing the signal onto other cells. There are many kinds of signaling molecules, and some of these are fat soluble and some are water soluble. So when a signaling molecule is fat soluble, such as a glucocorticoid, for example, it is able to slowly diffuse across that fatty cell membrane and then bind to an internal receptor to carry out an action and elicit a cellular response. But when a signaling molecule is water soluble, it can't just cross the membrane on its own, just like oil and water don't mix. So it needs a special extracellular receptor to transmit a message into the cell. That's where receptors come in. I just want to review the four primary receptor types. We have intracellular receptors, which, true to their name, exist inside of the cell, on or in the nucleus. These receptors react to stimuli by initiating gene transcription and then carrying out protein synthesis. There are three cell surface receptors. The ligand-gated ion channels react to ligand binding to carry out changes in polarization. This is seen in neuronal communication as well as in muscle tissue and at neuromuscular junctions, for example. The enzyme-linked receptors are enzyme protein receptors that carry out phosphorylation, gene transcription, and protein synthesis directly from the receptor. My focus today, unfortunately, is this, G-protein coupled receptors, which, as you can see, have a sort of wacky cascade of events occurring in reaction to a single receptor activation from a ligand. Since the GPCRs are the largest and most diverse group of signal transduction enzymes, there are about 800 GPRC genes which have been identified just in the human body. I will not be discussing all 800 in this presentation. Most of these are involved in sight, taste, and smell, about 500 of them, but a good portion are also associated with hormone detection, detection of growth factors, and other ligand binding processes. Phylogenetic analysis has revealed that GPCRs emerged from a common ancestor as long as 1.2 billion years ago, which is before plants, fungi, and animals, and they're the target of more than a third of all pharmaceutical drugs on the market. So I mentioned before that there are over 800 genes which code for G-protein coupled receptors, and like all the rest of biology, these receptors are classified based on their structure and function. There is class A, which is divided into the rhodopsin and non-rhodopsin receptors. Rhodopsin, as you may know, is involved in vision and detecting light. Some examples of the non-rhodopsin receptors are the adrenergic receptors and opioid receptors. My focus today will be on class A receptors because they make up about 90% of GPCRs. But there's also class B. These are the secretins. Class C receptors are the metabotropic glutamate receptors. And class F, which includes the frizzled and smoothened receptors, which some of which are involved in taste. And if you're wondering, there are classes D and E. These are fungal mating receptors and cyclic AMP receptors. But the structures and the processes of these are less understood than these other four classes. GPCRs are considered a superfamily of proteins, and as you saw in the previous slide, there are a lot of variations. There are variations to the specific amino acid sequences, which ligands they respond to, and their associated messenger proteins. But here's a rundown of the basic structure of the integral membrane receptor, its G protein, and its signaling pathway within the cytosol. So to get started, let's look at this sort of generic G protein coupled receptor diagram. Of course, as with all cell signaling, we start with a ligand, which is any molecule or signal that activates these membrane proteins to start a receptor pathway. The active or allosteric binding site in all GPCRs consists of 7 alpha helices, which cross the membrane and form a sort of basket shape within the phospholipid bilayer. So here we have 1, 2, 3, 4, 5, 6, 7. Each alpha helix is between 20 and 28 amino acids in length, and because of this feature, these are sometimes called heptahelical receptors or 7 transmembrane receptors. There is an N-terminal domain on the extracellular side, and there's a C-terminal domain on the intracellular side as well. And now if you look beneath this, you can see this gamma, beta, and alpha complex. These 3 units make up what we call the G protein. G is short for guanosine nucleotide binding protein, which is a trimeric, tri meaning 3 protein. So in its trimeric form, this G protein is affiliated with the GPCR because it has some lipid components which loosely bind to the transmembrane receptor. In its inactive state, this G protein is bound to a GDP molecule, but when it becomes activated by the ligand, it dissociates from the GDP and binds to a GTP molecule, which is abundant in the intracellular fluid. So once the ligand activates the transmembrane protein, it causes a conformational change and then releases these subunits into the alpha unit and this gamma beta unit. These secondary proteins are then free to elicit a cascade of messages within the cytoplasm. And I want to mention also that although it's not in this diagram, there is a secondary affiliated enzyme or effector protein, which is either transiently affiliated with the membrane or a membrane bound, which carries out the signaling pathway. So just to sum up here, there are 3 main components which are common to all G protein coupled receptors. First, there is always a plasma membrane receptor with 7 helical segments. Second, there is always an associated G protein, which cycles between active and inactive states. And third, there is always an effector enzyme or ion channel, which is activated by that G protein. So, okay, let's look a little more closely at one of the most well-understood GPCRs, which is the beta-adrenergic receptor system. This is a class A non-rhodopsin GPCR. Okay, here we have the glorious beta-adrenergic pathway, which begins with a ligand called epinephrine. Most people are familiar with epinephrine or adrenaline, and this is how it acts specifically in the body. So the first step is that epinephrine binds to this beta-adrenergic receptor. And once that happens, this protein, which up until now was bound to GDP, right here, it releases its GDP, which is replaced by GTP, and then enters its active conformation, meaning this alpha subunit is released. Once activated, this released alpha subunit, referred to as G sub S alpha, the S stands for stimulatory. So this is the portion of the G protein alpha which is going to stimulate something else. The next step is that this activated alpha subunit moves to another nearby transmembrane protein. And in this case, it is adenylyl cyclase. This alpha subunit then stimulates adenylyl cyclase to form cyclic AMP or CAMP from ATP. Once cyclic AMP is formed, it's now going to activate yet another protein called protein kinase A or PKA. Protein kinase A is the great phosphorylator. So pKa is now going to phosphorylate other cellular proteins, such as glycogen phosphorylase B kinase, which is the enzyme responsible for initiating the process of glycogen breakdown in the liver and muscle. It does this by targeting the serine or threonine residues. So this is essentially how epinephrine works to release energy in the form of glucose in response to an external threat or stress. So you might be wondering how that activated G protein alpha subunit gets back home. Well, G proteins have a really cool feature, which is that these proteins have GTPase activity built in. So after a bit, that G sub alpha subunit will just naturally turn itself off by hydrolyzing that bound GTP so that it reverts back to GDP. Once this alpha subunit is again GDP bound, it will find its way back home to its gamma and beta subunits and reassociate, once again forming a trimeric protein, G protein, until a new ligand begins that activation process again. But wait, there's more. All right, so I talked about how that activated alpha subunit makes its way back to its original trimeric form. That subunit has intrinsic GTPase activity, so it eventually deactivates on its own. This GTPase activity is fairly slow, however, so it can be helped along by an extrinsic GTPase activator protein called a gap protein. The abundance of these proteins within the cell varies based on other regulatory factors, and there are some other mechanisms involved in the deactivation of that epinephrine response pathway as well. The one shown in this image is a sensitization pathway when epinephrine is in abundance and just is constantly binding to that receptor. This process is mediated by beta adrenergic protein kinase called BARK and another protein called beta arrestin. So after epinephrine is bound to the beta adrenergic receptor and that trimeric protein is disassociated, not only does the alpha subunit play a role, but the other two subunits, the gamma and beta dimer, will recruit a protein called BARK or beta ARK or BARK to the membrane, where it will phosphorylate the serine residues at the inner carboxyl terminus. So after BARK has done its job here, beta arrestin will come along and then bind to that phosphorylated carboxyl terminal. And this is where it gets cool. The receptor arrestin complex is then completely engulfed and it enters the cell by endocytosis. So this whole thing enters a little micelle. So where it can't detect extracellular epinephrine. So this is how it's desensitized to epinephrine. Once this little inside vesicle arrestin eventually will dissociate, the receptor will become dephosphorylated, and then it'll finally return back to the cell surface as an active GPCR. So before I move on to another type of GPCR, I wanted to talk briefly about protein kinase A. I mentioned previously that this protein is responsible for phosphorylation of other proteins down the line of the signaling cascade. So this is what this protein looks like. It's composed of two identical catalytic subunits and two identical regulatory subunits. So you see these here. These are the regulatory and these are the catalytic subunits. I'm terrible at circling. This tetrameric complex is inactive because there is an autoinhibitory domain on each R unit that sort of blocks the active binding site on the C units. So when cyclic AMP is available, this activates pKa by binding to these R subunits, causing a conformational change which separates the two C and the two R units. So the C units, their catalytic subunits, separate entirely. You can see them here and here. And it's the C subunits which bind to ATP, and then transfer a phosphoryl group from this ATP to those downstream proteins at the serine or threonine residues. There are many CAMP dependent protein kinases, and they, like most proteins, vary greatly, but they all have a similar mechanism with this autoinhibitory binding site, which can be activated by cleavage of the subunits and then inactivated when they are once again reunited. And one other feature about GPCRs is that of amplification. So if you look at the bottom here, you can see that a single epinephrine binding event to a GPCR will yield about 100 million molecules of glucose into the blood. So this is through a signaling cascade involving, of course, the GPCR receptor, that active G protein, the effector protein adenylyl cyclase, protein kinase A, which is, where is it? There it is. And phosphorylase B kinase, as well as glycogen phosphorylase B activation into simple sugars, ultimately breaking down glycogen into simple sugars. I only have one other GPCR, and this one reacts with light. This next one is also a class A GPCR, and it's called rhodopsin. Okay, so this is rhodopsin. This is also a class A1 GPCR, but instead of binding to a ligand, it is activated by light. Rhodopsin, like beta-adrenergic receptors, is coupled with a G protein, but in this case, it's a protein called transducin. And that's right here. The first step in activation of this GPCR is light absorption, which is literally a single photon or photons. The protein component of rhodopsin is called opsin, which is right in the middle disc membrane bilayer. Once a photon is absorbed by opsin, it causes a change in an internal retinol molecule at the 11th carbon, making 11 cis-retinol a trans-retinol, essentially just straightening out. It's like this, and it straightens it out, this retinol molecule. So this tiny change causes the rhodopsin molecule to be able to interact with its own special trimeric G protein, which again is transducin. Once transducin is activated by the GDP-GTP replacement, it also dissociates into alpha and beta gamma subunits. The activated transducin then stimulates another membrane protein by removing its inhibitory subunit called cyclic GMP phosphodiesterase, or CGMP PDE. This activated protein then degrades another second messenger by reducing it in order to keep these calcium sodium channels open. What I think is so cool about eyeballs is that their membrane potential changes are sort of opposite to the other neural transmitting membrane potential changes in the body. So once again, cyclic GMP has been reduced. These cation channels close and the membrane becomes hyperpolarized. That's here. However, calcium is still effluxing out of the membrane via the sodium-calcium exchanger, making cytosolic calcium levels much lower inside the cell. So this reduction in calcium causes another protein to be activated. This one is called guanylyl cyclase, or GC. That's here. And this will inhibit phosphodiesterate, ultimately causing those original cation channels to reopen. And finally, we have another enzyme called rhodopsin kinase. This protein will be activated by those low calcium levels, and they'll stimulate the phosphorylation of rhodopsin right here. Apparently, arrestin likes phosphorylated proteins because now an arrestin protein, this one is called arrestin 1, is going to come in and bind to that rhodopsin protein. This leads to the reversal of that initial cis-trans-retinol change as the retinol molecule reverts back to its 11 cis conformation, that crooked conformation so that it is ready for another photon to begin the cycle all over again. Okay, because there are so many types of GPCRs, it stands to reason that there are many disorders associated with GPCR mutation or malfunction. There are more than 30 diseases which have been directly linked to GPCR problems, including central nervous system disorders, metabolic diseases, cardiovascular problems, respiratory diseases, gastrointestinal, immune and musculoskeletal problems, and even cancer. So I have listed here a few of the inherited disorders. TSH receptor mutations have been linked with certain types of hyperthyroidism. One of them is called autosomal dominant non-autoimmune hyperthyroidism. And the LHR gene mutations have been associated with precocious puberty disorders in males. Diabetes insipidus is associated with nephrotic GPCRs like the aquaporin 2 and V2 receptors. There is also retinitis pigmentosa. Some fertility issues are linked to GPCRs, and a certain bacterial pathogen you might be familiar with is Vibrio cholerae, which invades intestinal cells, locks the G protein in the active site, and causes extreme dehydration due to that constant nonstop ion efflux. And I have right here, I didn't mention this one. These are the autoantibody associated GPCRs, and there are more than I can even count here. But we have rheumatic issues, neurological issues, some cancers, endocrine, cardiac, and pulmonary problems associated with these. Drugs that target GPCRs have many potential applications and are already in use. I mentioned earlier that a third of the drugs on the market target G protein coupled receptors. Some of the possible uses include cancer treatments, managing diabetes and obesity. In cancer, for instance, these receptors help control tumor growth and spread by activating certain cell functions. There are opioid agonists, which target the mu-MOP receptors. Those are the group A non-rhodopsins, I believe. Antihistamines target GPCR histamine receptors. Many diabetes and multiple sclerosis drugs are GPCR agonists, they're activators. And drugs which target pain management, such as prescription opioids, target these receptors as well. It's not surprising also that most illegal drugs are GPCR specific too. The hallucinogens, such as LSD and psilocybin, are black market fentanyl and morphine, as well as the cannabinoids such as THC, all target various GPCR receptors. There are so many GPCRs, and it's impossible to review them all in one video, but I hope this provided a decent overview of the ways GPCRs work in signal transduction. So just to wrap all of this up, here are the key points once again. GPCRs have 7 alpha helical regions which cross the membrane, and within this 7 transmembrane basket is the allosteric or active site. Once activated, the site binds with a G protein, which will then release GDP and bind to GTP. The active alpha site will then act with another effector protein, such as transducin or adenylyl cyclase, though there are other effector proteins in different types of GPCR signaling. The effector protein signals another protein called protein kinase A or PKA to enact a cellular response. And many times this results in amplification response due to a single ligand binding event. This mechanism can be deactivated either by helper proteins such as GAP, arrestin, or BARC, and then the associated G proteins once again return to their original trimeric conformation. GPCRs are involved in many biological processes and virtually every tissue type in the human body, and when they malfunction, this can cause disease. This also makes GPCRs targets for many pharmaceutical and, unfortunately, many illicit drugs. And most importantly, let's go over the 3 commonalities that all GPCRs have. All GPCRs consist of a plasma membrane receptor with 7 helical segments crossing the membrane. They are all associated with a G protein, which cycles between active and inactive conformations. And finally, all GPCRs interact with an effector enzyme or ion channel, which is activated by the G protein. Here are my references. Thank you.